All right. Hello, 12s. We're continuing on talking about eukaryotic cells. Today, what we're going to discuss is the other membranous organelles. So we talked about the nucleus that was surrounded by that double membrane. We talked about the endomembranous system, which was the outer membrane of the nucleus, the rough endoplasmic reticulum, the smooth endoplasmic reticulum, and the Golgi body, and then the lysosomes, endosomes, and secretory vesicles, transitional vesicles. Those are all organelles. Those are all organelles used to make proteins and enzymes and export proteins and enzymes, excrete them, as well as lipid molecules, right, made in the smooth endoplasmic reticulum. So hormones and cholesterol and phospholipids made in the smooth endoplasmic reticulum. So that's all the transport system. Also, partly the recycling system. The smooth endoplasmic reticulum takes broken down stuff and sucks it up and cleans out the cytoplasm. Those lysosomes and endosomes destroy old organelles and also eat stuff from the outside of the cell. So that's all that stuff. There are two more other membranous organelles that we're going to talk about, and these have to do with energy, with creating energy for the cell so that it can do all those other things like DNA replication and protein synthesis and enzyme synthesis and lipid synthesis and so the first one we're going to talk about is mitochondria. You will find mitochondria, it's known as the powerhouse of the cell. You'll find mitochondria only in eukaryotic cells, both plant cells and animal cells and fungal cells. And mitochondria also has an outer membrane and an inner membrane, like the nucleus, except the inner membrane is all folded up. And it's folded up into these things called Cristae. And the folding of the inner membrane, the advantage of that is it increases the surface area. If it was just an inner membrane just on the inside, it would only have the surface area that was the inside of the mitochondria. But it being all folded up in there, that increases the surface area. We have a lot more membrane as a result. And the reason for that is you need that membrane all folded up because producing ATP, which remember is the currency of energy in the cell, happens on that inner membrane. So the more inner membrane you have, the more space you have for the proteins and the enzymes that sit in that inner membrane that produce ATP, so you can produce more ATP per mitochondrion. So let's look at mitochondria. So here we go. There is mitochondria like that. There you go. And there's the inner membrane all folded up. These are the cristae. The folds are called the cristae. The inside is called the matrix. And what happens across that enzyme, across that membrane is, remember when we talked about metabolic pathways, a glucose molecule in the cytoplasm undergoes glycolysis, so it gets broken down. And the net effect is 2 ATP from glycolysis and two carbon sugars. And those two carbon sugars are called, let me just, basically they're like acetate ions. They're two pyruvates or whatever, that there's these two carbon sugars that get made. And then those two carbon sugar, they just have 2 carbons, that's all that means, is they go into the mitochondria and they get smashed and broken apart across that membrane of the mitochondria. And they undergo what's called the electron transport chain and the Krebs cycle. We're not going to worry about the details of what happens in the Krebs cycle and the electron transport chain. That's the word I'm looking for. We're not going to worry about the details. All you need to know is that it just happens on that membrane. You can look in the textbook. I know they have a drawing of it somewhere in the textbook, but don't worry about the details of it. I'm going to ask you about the details of it. You just need to know that it happens on that Cristae membrane. On that membrane, the more membrane you have, so the more folds you have, the more efficient, the more you can carry out, the greater the surface area. And that process produces 36 ATP. And remember, we talked about that in the last unit. So eukaryotic cells, for a single molecule of glucose, make 38 ATP, thanks to the mitochondria. Anything else? Oh, how could I forget? So mitochondria have something amazing also in them. They have DNA. Mitochondria have DNA. So you'll find DNA in the nucleus for all the proteins and things, and then you'll have mitochondrial DNA that codes for the proteins and the enzymes in the mitochondria. And you inherit the mitochondrial DNA actually from your mom. We all have our mitochondrial DNA from our mothers. So when they do like assays of inheritance and stuff like that, they can look who your mom is by looking at your mitochondrial DNA. The advantage of having mitochondrial DNA is if your cell all of a sudden needs lots of energy, and therefore it's getting a lot of sugar or something like that. Instead of going through the whole DNA, mRNA out of the nucleus into the endoplasmic reticulum, ribosomes here, this and that, and boom, 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 all of that endomembranous system, instead of doing all of that, just run that mitochondrial DNA right there. You've got mRNA, you'll get a ribosome in there, boom, and you can make extra mitochondria, just the mitochondria, really quickly if you need them. So, boom, you got mitochondrial DNA. Eukaryotic cell genesis theory, that mitochondrial DNA, most likely those mitochondria used to be a prokaryote that another prokaryote, a larger prokaryote, ate. And over time, that smaller prokaryote evolved, that smaller bacteria, they were talking about bacteria here, evolved to become the mitochondria within the larger eukaryote. We don't need to go down that whole road. But essentially, that's how eukaryotic cells most likely came about. And they produced a lot more energy, which allowed the cell to make the other organelles in the nucleus and other things. Okay, so I feel pretty confident that's what you need. So you got glycolysis, and then Krebs cycle, and then the electron transport chain happens on the membrane of the cristae. So glycolysis happens outside in the cytoplasm. Krebs cycle, electron transport chain happen across the membrane of the cristae. Glycolysis produces 2 carbons outside there, and those 2 acetate ions, or whatever they are, the 2 pyruvate. I always thought there was pyruvates. Those 2 carbon sugars. And you guys can look it up. You'll know better than I do. Those 2 sugars enter in the mitochondria and makes additional 36 ATP. I hope that makes sense. What else is if the mitochondria? So if you make extra mitochondria, then you don't need that many. You don't want them sitting around running, taking up resources. They get broken down by those lysosomes. So endosomes that turn into lysosomes, they can be broken down, recycled back into the smooth endoplasmic reticulum. Anything else I want to say? Well, here's the cool, you guys will see it in the notes, but there you go. This is what an actual mitochondria looks like through electron microscope, micrograph. It's been sliced. You can see there's the cristae, the folds in there, and the matrix fluid inside. That's some rough endoplasmic reticulum there, if I ever saw any. And you see that it's close because this produces energy, and that energy can go right to the endoplasmic reticulum, used to assemble amino acids into proteins. Okay, the next membranous organelle is called a chloroplast, and you will find chloroplasts only. You will find that. I mean, you'll only find this in plant cells. Let's just say we only find it in plant cells. not in animal cells, not in fungi cells. So you'll find it in plant cells. And essentially what happens in chloroplasts, well, let's just look at it here. You have these things called thykaloids. That's these kind of pancake things, and they're in these stacks called grana. This is also to help, increase surface area. And it also has a dual membrane. It's got an outer membrane and an inner membrane, and there's, fluid inside. And so what happens inside these chloroplasts is there's two processes kind of happening, and here they are. So what you get is inside these thykaloids, light from the sun or from lamps and water come together in here. The water, basically there's a special pigment called my mind is going, chlorophyll. There's a special pigment called chlorophyll in here. And what happens is the light charges up the pigment, the chlorophyll pigment. And what it does is there's an electron in that chlorophyll pigment that is orbiting around one of the atoms. And it gets excited. The light hits it, and it basically orbits faster, and it raises to a excited state. Details of it, not important. It raises to an excited state. And then it crashes back down. And when it crashes back down, it releases energy. And that energy is used to wrench, to break apart water molecules into oxygen. So that's where the oxygen comes from, why plants breathe oxygen. And to make an ADP and an NADPH. And then that ATP is used in the Calvin cycle, so the plant is taking in CO2 just like we do. And that stuff is used to make sugar. So first you have the light reactions, and then you've got the Calvin cycle, which produces sugar. So plants make their own food, and then that sugar can go to the mitochondria in the plant and be used to make more ATP. So basically the light reaction produces ATP that will then power the Calvin cycle that will produce sugar that will then be used to make ATP for the plant. And imagine that sugar will not always be used. That's important. Will not always be used. That's not in the notes here. So this is like the secret info. Remember this. It will not always be used for energy. It will not always go to the mitochondria. That sugar will also be used, the carbons in it and stuff, it'll be used and assembled to make starch. So the plant will put the sugar into storage. So like rice or something like that, that's made out of starch. It's a very starchy. So that's it basically, and that CO2 that is assembled to make that sugar comes from the air. So think about rice, the sugars in it are actually the air converted into rice. So that sugar it'll be used as storage, as starch. It'll be used to grow more plant. The plant is made out of sugars, basically the stalk and the green and the leaves. So that plant is again air converted using sunlight, the energy from the sun, into plant that then you eat. It's pretty cool. How much detail? Don't get into too much detail. Just know that these things happen. The review questions are a little bit detailed. If so, do them. But anyways, I don't want to say too much about that. I think that's good. So chloroplasts are only in plants. Inside the chloroplasts, inside those grana, which are those stacks of thykaloids, what you're happening is you're having light reactions and you're having the Calvin cycle. So you got two processes happening. The light reactions take water and light and convert the water into oxygen and energy. Then that energy is used in the Calvin cycle to take carbon dioxide and turn it into sugar. So that's the best thing to know. And then that sugar is used as storage, as structure for the plant, and as energy for the plant to do stuff. Is there any more that I want to say? It might be just that just might be everything that I need to say. Oh, about that electron. So the light, just to help you understand that, the light. So how does the light excite the electron? What does that mean that it excites this pigment, this chlorophyll pigment? Well, if you've ever had, those glow stars on your ceiling in your bedroom, or have ever seen a glow, a toy that you charge up with light and it starts to glow, that green glow, around Halloween or whatever, we have lots of toys like that. And actually, if you shine UV light on those toys, they will glow a lot brighter, because they are covered in a special pigment like chlorophyll, except what this pigment does is it takes, so these electrons in that pigment get excited by the UV light, so by the sunlight or UV light, they get excited, they raise to that next level, and then they slowly, over time, they fall to another level, and basically what they do is they get excited by the UV light, and when they fall, they release energy, they release that glow, they release it as visible light. So basically they're converting UV light into visible light. That's essentially the same thing that's happening here. Is those electrons, they get excited by the light, they absorb the light energy, the photon energy, and then they fall slowly, and as they fall, they release energy that is used to break down water into oxygen and ATP. Okay. I think we're good. That's other membranous structures inside the cell.